I think we have most people on the call that are going to be joining us. If they continue to arrive in this session, that's fantastic. But I want to thank you all for joining the Canadian Association of Agri-Retailers Care Connect sessions. My name is Myrna Grawn, and I'm the Executive Director of CARE. And if we could move on to the next slide there, Jordan, I can just want to share a little bit with you about CARE. Our vision is to truly be that successful and thriving Canadian agriculture retailers and farmers. We represent, as a member association, all of the ag retail in Canada, and there's about 1,600 of them across the country. Our mission is that CARE is the united voice for the Canadian ag retailers, advocating for responsible agriculture and fostering industry growth and sustainability. So as we continue on here, I just want to do a quick touch base on who the team is. If you need to reach out to any of us, Jordan, you'll meet shortly. He's our Marketing Industry Relations Manager. We have Diana Svensson, who's our Office Admin Coordinator. So anything that you need, you can call our office, and Diana will handle and sort of quarterback a lot of the questions you may have. Lisa Siragusa, who's not able to join us today, she is our Member Relations and Training Manager. Tracy Shelton does all of our communications and conventions. So if you're planning to attend convention this year, it's in Regina, and it's November 17th to the 19th. We have Erin, she's our controller, and Juliana is our summer student working on all of our CRM and different membership distribution directories and keeping those all up to date. Our board of directors, we just had a week of board meetings, and so you'll recognize some of those names. They are from across mostly Western Canada. We had our 2 recent retirements from the board, and that's our representation from Ontario and BC, and we're going to be looking at filling those positions. So if you know of anyone who's interested, you might want to give them a little nudge to see if they'd like to join our board. So if we move on to the next slide, I do want to mention we are located in Winnipeg. If you would like to become a member, you can just handle this QR code with your phone while it's up on our screen here. However, if you just go to our website, there's there's Join CARE. We have our social media that you could follow us on as well, and you can see the 5 different platforms there. So our next session, so today's is starting shortly, but our next session is going to be Kirsten Hannum, and she's going to be discussing growing tree fruit under water pressure, and that's tools for drought resilient orchards. So that might be something of interest, or if you know someone who needs to maybe hear that one, you could forward them the sessions when we promote this one. All right, and now over to you, Jordan.
Perfect, appreciate it. Just for the fun housekeeping side of this, remember, as presentations going on, just make sure that you're muted so that we don't get any of those awkward background conversations. And also, feel free, your choice. We love seeing smiling faces, and it helps motivate our speakers as they go to know that eyes are locked in on them and that they're actually making ground and headway with understanding. So again, feel free either way to turn that camera on, or as we see most of you keeping it off. But that is it for the housekeeping side of things. We are very lucky to have our speaker, Dr. Barbara Caidmanan. She is a soil scientist whose research focuses on phosphorus cycling in soils and water. Her work examines the forms of phosphorus present in agricultural systems and the process that influence phosphorus availability, movement, and environmental impact. Her research contributes to improved understanding of nutrient management strategies that support crop productivity while reducing the risk of phosphorus loss to surface waters. So without further ado, I will stop sharing our screen, and we will let this get underway.
All right, let's start sharing my screen. All right, that seems to have worked. Everyone see my screen? And can everyone hear me? All right, good. All right, as said, I'm Barbara Caidmanan. I am based in Swift Current, Saskatchewan, and I'm going to talk about some of the research I've done while I've been here. All right, well, my slide presentation is not Oh, there it is. So why do we study phosphorus? It's an essential element for all organisms, people, everyone. It's part of our DNA. It's part of what gives us energy for our ATP. Without it, we can't live. In agriculture, we study it because the too little bioavailable phosphorus is going to affect our crop yields. But we also are concerned because too much is a well-known factor in water quality, as this famous picture shows from David Schindler's study in the 1970s in Ontario when he added phosphorus, carbon, and nitrogen to one side of a lake and carbon and nitrogen to the other and triggered algal blooms. But there are many different kinds of phosphorus compounds in soils and the environment, and they vary in their bioavailability and reactivity. And so we need to study them to understand what's there and how to manage it better. So the background of my talk is such that I'm going to talk about phosphorus forms and cycling and methods to study phosphorus. I'm going to talk about some of my research focusing more on some long-term studies, some agricultural land use studies, and some loss and runoff, and then a summary. I will note there's a lot of text on my slides because I know people can sometimes have a hard time understanding my voice. But all right, so we talk about phosphorus. Most of you are familiar with phosphate, which has either dinitrogen phosphate or one nitrogen, or we can also have something we call orthophosphate. This is where there's no hydrogens on it though, and this is a special case, and I'll get to it a bit later. But this is not the only inorganic form of phosphorus. We can also have pyrophosphate and polyphosphate. These are chains of phosphorus. Pyrophosphate specifically just has 2 phosphates, but these chains can be very, very long, and they are quite natural. There's often storage compounds for organisms. When we talk about protons, as I hear, the hydrogen or protons, they dissociate or become removed from the phosphate groups at different pHs. So below pH 2, all the protons are there, but at 7.2, it loses first 1, and then it loses. And then as we get up to over pH 12, there's no hydrogen associated with it. And this particular case is orthophosphate. And again, at soils, we tend to pH of soils from 5 to 8, we're going to have dihydrogen phosphate or hydrogen monohydrogen phosphate. Orthophosphate isn't present. Some people call it that and call it all phosphate orthophosphate, but that's actually not the case. We also note that below pH 5, phosphate tends to be associated with iron, aluminum, and manganese, but above pH 5, it's calcium and magnesium. For most of you, the inorganic phosphorus you're used to thinking of are fertilizers. So monoammonium phosphate, which has ammonium plus phosphate, diammonium, again, 2 protons here, 1 proton here. Ammonium polyphosphate, it's a liquid fertilizer. We can also have super triple phosphate, which is much more common on the coastal parts of Canada, whereas these are more common on the prairies. And then even struvite, which is sold as crystal green, which has magnesium phosphate with ammonium. And this is actually precipitated from wastewater and sold. It's actually a recycled product. Most of you, though, tend to think of phosphorus. You'll think of it as sort of being for example, MAP as being 11520. And this is labeled this way because it says it's 52% P2O5. Now, what you may have noticed is this is the first time I've mentioned P2O5. And I put all of these equations up here because if you look really carefully, none of them contain P2O5. In fact, it's impossible for any of them to contain P2O5. P2O5 dates back, the term P2O5 dates back to the 1800s when nutrients were thought to be salts. Now we know they are not. Strangely enough, in the lab, my lab, but even any other lab, if you send for soil testing, they measure nutrients as phosphorus, not P2O5, and then convert them to P2O5, which is a compound that simply does not exist. Somebody needs to be muted, please. Anyway, it does not exist. We need to stop using it. Let's get rid of it. Let's just start putting P on fertilizer bags. We can include the P2O5, but also put P and just gradually get rid of it. It does not exist. Anyway, have to get off my soapbox for that. The other there are lots of organic forms of phosphorus as well, and these are where phosphorus is bonded to carbon. Now, I don't show the carbon in these, but at each of these corners here, think of that there's a carbon group. And so you will see there's a phosphate group associated. And we divide organic phosphorus compounds into how they're bonded to soil. So phosphate monoesters have a single ester linkage. That's one oxygen to carbon. And these include some important compounds like inositol hexachous phosphate, IHP, which includes phytate, which is a storage compound in grains, and is also often fairly important for nutrients, particularly human and animal nutrition. They also sugar phosphates. The next category are phosphate diesters, which have two ester linkages. So again, there's carbons where each of these benzynes are. These include phospholipids, these RNA, DNA, so the molecules of life. And then the last major compound are what we call phosphonates, and these actually have a direct carbon-phosphorus bond. These are often, for example, glyphosate is the active ingredient in the herbicide Roundup. Sorry. So some of them are synthetic, but they're often quite natural. Here, phosphomycin is actually an antibiotic produced from streptomyces, which are soil organisms, and it's used to treat things such as tuberculosis. So again, many different ranges, and these are all important, and I see all of them in all sorts of soils and environmental samples. So why do the forms matter? Well, it affects their cycling and availability. And just to show, it depends on the bonding type that you see here. For example, there's no charge associated, but a compound like phytate with lots of phosphates will bind tightly to soils. Polyphosphates have a charge, but things like phosphatidylcholine here, which is a phospholipid, don't. There's only one place that phosphate could bind to soil. So all of these are going to affect, but on the other hand, these have lots of carbon, so microbes might attack them for the carbon or nitrogen. For processes controlling soil phosphorus cycling are first precipitation and solubilization. This is where, say, a phosphate group binds with calcium to form a calcium phosphate or an iron phosphate, and then that's returned back by solubilization. We also have adsorption or desorption. So this is on a mineral surface in soil. So the phosphate or other compound is going to bind to the minerals. It's not actually in soil solution. These are what we refer to as chemical processes. But we also have biochemical processes, and these include uptake and immobilization, where a microbe or a plant takes up phosphate from the soil solution, because that's what it uses, and it can either store it as an inorganic phosphorus form, like phosphate or polyphosphates, or it converts it to organic forms. It converts it to DNA or cell wall phospholipids or ATP. And then these return back by decomposition and mineralization. And for the people more familiar with carbon and nitrogen, in phosphorus, decomposition and mineralization are different processes. Decomposition, as say plant residues decompose, they're going to release both phosphate and organic phosphorus, and often indirectly, because whatever's decomposing that organic matter is probably more interested in nitrogen and carbon than phosphate. But then mineralization takes the phosphate from those organic compounds and releases it, and that involves a very specialized set of enzymes. So for example, for phytate, it takes phytase. So all of these processes affect both organic and inorganic P compounds. So DNA and phytate can sorb, precipitate, for example. So this is a model I drew for a few years ago for a book chapter showing phosphorus cycling. So these are two soil particles, so a root and a microbe. So we're right at that very fine scale, and we can assume that we have on this side it's all the inorganic processes, and this side is the organic. So we can have sorption to the soil particles, organic and inorganic. We can have precipitation and solubilization. We can have uptake by roots or microbes, release of our dissolved organic phosphorus, which also absorbs precipitates, etc. So if we have our phosphate fertilizer and we put it into the soil solution here, it has the option of being sorbed to the soil particle. It has the option of being precipitated with cations in solution like calcium or aluminum, or it could be taken up. The point to recognize is all of these processes occur simultaneously. So there's a lot of competition going on for that phosphate that ends up in soil. And also, again, to note that these processes affect organic and inorganic compounds. We talk about soil phosphorus pools. We have to note that phosphate transport is diffusion limited and organic. So it moves into the soil solution. So you start, if you put the fertilizer in, it will move away from that fertilizer based on concentration from high to low, but that depends on temperature and moisture. So in dry soil or cold soil, we often see what looks like phosphate deficiency, purpling, but that's just because it's not moving because it's cold. It's a diffusion thing. It's not necessarily any kind of a deficiency. So we talk about pools. We have what we call the labile pool, and that's what's easily moved into solution. That includes both phosphate and organic P compounds, moves by diffusion. If it is organic P, those are the ones that are like things that turn over really fast because something's going after carbon or nitrogen. We have moderately bound, so that's where it's more tightly bound. It's sorbed onto the minerals. It's harder to get. It needs some additional reaction to remove it, not just by diffusion. And so plants have strategies to get this, and I'll talk about it. And then finally, we have tightly bound. This is almost impossible for plants to get. Sometimes it's what we call, it's blocked, and this again includes both organic or inorganic phosphorus forms. Most of you might be familiar with soil test phosphorus. That's where your lab gives you concentrations, and that measures the labile phosphorus pool. Say plants and microbes have strategies to access phosphorus. They can, for example, different rooting strategies with different volumes and different access. They will alter phosphorus chemistry. So a lot of microbes or plants produce organic acids that will desorb precipitated or solubilized precipitated phosphorus or desorb it, or they'll sometimes change the phosphorus chemistry just by changing pH, which will change availability. They also produce enzymes, both microbes and plants. Again, I talked about those that mineralize organic phosphorus, or there's the indirect because it comes from decomposition, is releasing phosphate from organic matter. And of course, they can make friends. A lot of microbial symbioses, particularly the mycorrhizal, involve phosphorus, or we can have associations of phosphorus solubilizing bacteria in the rhizosphere. So now we get into how we study phosphorus, and it depends on what we want to characterize. Do we just want to know general phosphorus pools, soluble air, or do we want to predict some kind of fertilizer response or environmental risk? And so for most of those, this involves some sort of extraction of a soil. So you can see it here, there's filters, and below that is a solution, and then we can measure it colorimetrically. So this color, the darker the blue color, the higher the phosphorus concentration. Or we can use advanced techniques. This is called an ICP OAS, or inductively coupled plasma optical emission spectrometry, and this will measure total phosphorus in solution. But if we want to know specific forms like phytate or apatite, I use some advanced techniques. Most people won't do this, but I try to use this to inform the results that come out of the simpler techniques. So one technique I use is NMR spectroscopy, and this is where we put the sample into a big magnet, and this gives us a spectrum here, which you can see all these different compounds I talked about. Phosphonates here, DNA, pyrophosphate, polyphosphates. This is equivalent to an MRI at the hospital that works about the same way, but is specifically for phosphorus. This is at the University of Saskatchewan where I do my work. I also have used, this is the Canadian light source, and this gives us, synchrotron analysis, again, gives us spectra with different compounds, but it also allows us to map. So phosphate here is associated with calcium or iron or other elements. Again, very advanced techniques done on a very different scale, not routine analyses, but they help inform our other analyses. So when we talk about analyses, what do we measure? Well, total phosphorus is obviously all the phosphorus is sample. We do this by some sort of hot acid or digestion. I measure it colorimetrically. Total organic phosphorus is all the phosphorus associated with carbon or organic matter, and this is usually measured by ignition and extraction. The soil test phosphorus I've talked about, this will depend on your region as to what you use. Olsen P is with a bicarbonate extraction. Bray P is usually from our acidic soils. Same with Malic is fairly common in the eastern Canada. Kelowna P on the prairies. It's important to recognize, though, that these were not designed to predict plant available phosphorus. Rather, they're designed to predict crop response to phosphorus fertilizers. So they need to be calibrated. And so a concentration of 10 ppm or 10 milligrams per kilogram of phosphorus in some soils might be high and some might be low. You need to calibrate it to understand. We also, again, talked about those pools, the fractionations. We can measure water-soluble phosphorus. That's an environmental test. We can measure microbial, or as I said, if we want to look at specific forms, we use the really advanced techniques. One thing, though, to understand about the soil test phosphorus that, again, is what many people are going to see from a commercial lab. Total phosphorus, this is an example from a Saskatchewan soil. Total phosphorus is 1,000 ppm or milligrams per kilogram. Those are the same thing. In my soils in Saskatchewan, most of them are 50% organic phosphorus. So half of it. Soil test phosphorus is maybe 30 ppm. It's a minuscule proportion of the total phosphorus. But again, for many people, that's the only phosphorus you will ever measure in your soils. Again, I talked about this very complicated phosphorus scheme. This is all you're measuring with soil test phosphorus. You don't measure any other pools. And so the majority of phosphorus, we don't measure it. We don't understand it. And this is what I focus on, is trying to understand. I don't do fertilizer trials, really. I'm interested in everything else for phosphorus that's there, not just the soil test P. The other important factor for phosphorus to understand is that it is stratified in soils. Even with agriculture, there's a cropland versus pasture land. It's always higher at the surface, always. And even with some tillage, you're mixing in here, but you're still not going to make it. It gets much lower for soil test phosphorus, at least, at much lower depths. And so this is really important as you visualize and understand where plant roots are or what's going on. And again, how you sample makes a big difference. If you sample the top where you're putting your fertilizer, you're going to get a very different concentration than if you sample down to 30 or 40 centimeters and mix that all together and analyze it. This is really important, especially for runoff loss. Pardon me. All right, so deeper sampling will dilute it. Excess P at the surface is a risk for loss. And so sample at depth of fertilizer placement. So now we're going to move into a bit more of some of the work that I do. So the questions that I ask in my research are, what are the forms and concentrations of phosphorus in soil with land use? With management practices, how do they change? How does phosphorus move from agricultural land and runoff, and what are management practices that can change that? So for those of you outside of Saskatchewan, I'm here in Saskatchewan. In particular, I'm based in southern Saskatchewan, so here in Swift Current. Saskatchewan's up here, Regina's over here. And so some of my work, I'm going to just talk about in this presentation, work from different parts of the province. And talking about some long-term fertilizer trials, a land use trial, and phosphorus and snowmelt. So starting with the long-term fertilizer trials, we have something we call legacy phosphorus. Legacy phosphorus tends to be what's accumulated from prior land management. So often when you add fertilizer to soil, most crops don't use what's added that year, so it builds up over time. Also though, of course, if you're spreading manure as a disposal mechanism, that will build up as legacy phosphorus. All sorts of different things will happen. And if that phosphorus is available to crops, which is the big question because it's not measured in our soil tests, it could also help to reduce the need or change how we view phosphorus fertilizers. It also, of course, contributes to loss and runoff, so we worry about it. And again, we need long-term studies with phosphorus buildup and drawdown to understand how it changes long-term. So these are some plots, a long-term study in Swift Current. It was established in 1967, and you can see there's a whole bunch of different rotations, as you can see here, and 3 replicates. I'm, for this, just going to focus on the continuous wheat. So that's wheat grown every year, with and without phosphorus. So for this study, we initially, again, here are the different, it was ammonium nitrate and urea, for ammonium nitrate initially, now to urea. It's always received MAP. And then in 1995, they stopped fertilizer on these, phosphorus fertilizer on these plots. And that in this, you can see the small quarter of the plot here. That was actually done not because of interest in phosphorus, but it was interest in cadmium, which comes in with a lot of phosphorus fertilizers, and wheat can be sensitive to it. We don't use them for that anymore, though, because when I was hired in 2008 as a phosphorus expert, these were fascinating to me. So nitrogen applied based on soil tests. These are also tilled, and all the wheat straws returned to plots after harvest. So the first thing we did was we just looked at the plots receiving N and P, and we looked at the plots from 1995 when fertilizer is applied, and then we looked at the plots with and without fertilizer. And so Olsen P, Malick P, and this is the top 0 to 7.5 centimeters of soil. And as you would expect, you stop adding fertilizer, it does, after 15 years, it draws down some of the phosphorus concentrations. I will note, though, that these are still reasonable, and we saw no yield differences whatsoever. These are, because it's continuous wheat, these are a bit lower yielding. And it's also important to remember, for those of you not on the prairies, that moisture limits everything first, and then nitrogen, phosphorus is usually a third. It comes after that. But no significant differences in yield. Again, when we looked at total phosphorus, and these are 2 depths, 0 to 7.5, 7.5, 15, so 0 to 3, 3 to 6 inches. Total phosphorus doesn't change. Organic phosphorus seems to increase, but that's because we're drawing down the inorganic phosphorus. The crops are taking that up when we're not adding inorganic fertilizers. We also used the Canadian light source and the advanced techniques and showed that it's actually changing the forms of phosphorus as well. And that made no sense to us. It can be drawing it down, but why are we shifting? Suddenly there's no calcium phosphates, or we're changing the kind of calcium phosphates. Hydroxyapatite is a calcium phosphate, but we're seeing these shifts in iron phosphate and aluminum phosphate. So that was a little odd to us. So we followed this up with a study in 2016. And in that case, we included the plots with and without nitrogen. Same thing, and then with and without phosphorus. These are the subplots. And same thing, no yield differences. There are some differences, but nothing statistically significant. This was interesting to us. This is soil pH, and as you can see, with nitrogen, the pH is about 5, and without, it's closer to 7. This is huge. This is acidification, and this is a cumulative effect of ammonia addition on the prairies. And we've seen this. We just published a paper on this. It's being seen in Alberta. This is a huge issue because, as you see, it affects a lot of things in soil. In particular, other cations. So again, as the pH goes down, aluminum and iron go up, and calcium and phosphate go down. And aluminum in particular is an issue because as you drop below pH 5, aluminum toxicity starts to appear, and it's already being reported in Montana. Again, we tend to think of our soils as being high pH or neutral pH, and they're not because of the ammonia we've been adding. Again, all correlates to pH. When we look at phosphorus, total phosphorus is starting to draw down. And again, though, it corresponds to whether or not you're adding fertilizer. Same with the soil test phosphorus, Olsen P, it corresponds to whether you're adding fertilizer. Organic phosphorus does not. It corresponds to pH. So not only are we changing just by not adding fertilizer the drawdown, but the pH change is affecting all the other phosphorus that's cycling in there and how that could potentially be bioavailable. And we also did a lot of studies and things. Again, this has been published. I won't go into detail. But to summarize these long-term studies, we didn't see any changes in yield or grain P from stopping phosphorus fertilizers, suggesting that plants can still access legacy phosphorus in soil many years after we stop fertilizing. Adding phosphorus to no phosphorus plots, though, actually significantly accumulated phosphorus, mostly because they were stressed for nitrogen. And so the phosphorus stays there, and that's an increased risk of P loss. Long-term addition of ammonia fertilizers increases pH change. It's a half a pH unit per 1,000 kilograms per hectare. This has been shown around the world, and that has changed the soil microbiology and soil chemistry. We have other studies to show this, although I won't talk about it today. With respect to soil acidification, as I mentioned, it's being widely reported. We need to start consider liming fertilizer tillage. It's actually worse. Acidification is worse under zero till because we don't spread that fertilizer. It really builds up. A depth of monitoring is important. So these are this is from a different trials that's called the 0MC. So with and without tillage, also continuous wheat. Soil pH, so the blue is no till. You can see it's significantly lower than with tillage and significantly decreases at the depth of fertilizer placement, as does exchangeable acidity. If you're soil sampling and measuring pH, if you separate it out, you'll see this, but if you sample it all together and mix it, you will not see this significant change. Why is this important? Because you're putting your seed right into this acidification zone. It's growing right through it. If you're inoculating with rhizobium, it has a very strict pH requirement of 6.5, which is not getting where there's acidification. This is something that matters. It's secondary to what I'm going to talk about now, but something I wanted to stress, and it was important. Anyway, I've got a lot of publications. I'm skipping through those now because if this is being posted, people can go back and find it. Now I'm going to move, in interest of time, I'm going to move into my land use study. Again, different land uses in Southwest Saskatchewan. We have native grassland. We do still have patches of it. It's managed and grazed. We have tame grasslands. These were planted in the 1930s during the Dust Bowl era. They're all crested wheatgrass. They're interesting because it's a single crop, or single plant species across. They've been maintained so for 70 plus years. We also have, of course, annual cropland, and we also have roadside ditches, which if you ever drive across the prairies, you know, those are a significant component of the agricultural landscape. And so this study was designed to look at all sorts of aspects. But my particular involvement with the study was to look at phosphorus in detail. We actually had blocks of these that were, you can see all four within a few kilometers, all four land use types, so very close together, which removes any kind of differences in terms of climate or soils. We sampled them, initial sampling where we actually did all the different depths, and then we had multiple analyses just using 0 to 30 centimeter depth. I've done all sorts of analyses. These papers are published. I'm just going to talk about, first of all, by depth. And this is what I showed you before, but I didn't have the roadsides on there. You can see significant stratification. And these are significantly different. Drops way down with depth for Olsen P. We also saw significant changes in bulk density, clay content, total carbon. Didn't affect total phosphorus at each depth. When we look at the 0 to 30 centimeters together, though, we do sort of get rid of some of these changes in Olsen P or Malick P, just because, as I said, you mix it all together. But there still are differences. This is where I start going back to talk about all those different phosphorus forms I said I looked at. So this is an organic phosphorus that includes phosphate and polyphosphates and such, the monoesters, the diesters, so phospholipids, etc. And then these are the inositol phosphates. So that's phytate and its other stereoisomers. And these are an interesting group because phytate comes from plants, but the other forms of IHPs come from microbes. And as you can see, we see significant differences in them, but we also saw significant differences in the forms, suggesting that the soil phosphorus comes from different sources under different land uses. This also ties back to soil enzymes. So acid and alkaline phosphatase will release phosphorus from these monoesters. Acid phosphatase is assumed to come from plants and microbes. Alkaline is thought to be from microbes. And so again, significant differences in land use and the biochemical cycling of phosphorus, or in diesterases for the diesters. We also saw significant differences in the microbial pools. So tying back to all of this, again, I'm shortening what was a very complicated study into a few minutes. But again, we saw significant differences with land use, especially compared roadsides and croplands compared to tame and native pasture. The differences were altered physical, chemical biology, soil pools and forms, and soil biogeochemistry associated with phosphorus cycling. Associated with degrees of disturbance. Again, roadsides are disturbed. Things move in and out of roadsides, ditches, tillage, crop removal, inputs from fertilizers, and even from manures, because these were grazed grasslands, different types of plants with different rooting depths. All of these things are important with respect to phosphorus in soil. Again, publications. You can look these up on when this is posted. So next, finally, disinterest time, I'm going to talk about runoff. We're concerned about runoff because of water quality. One of the things we're interested in is physical forms. Is it dissolved or is it attached to particles? And this is separated by filtration. Again, it affects transportation process. Something that's dissolved in water can move a long way, whereas a particulate is gonna change because it's a solid. It can't move in the same way. We're also interested in phosphorus concentrations and forms because that affects reactivity and bioavailability. And of course, on the Canadian prairies, we have a lot of winter. In fact, it's snowing right now, but although it's supposed to, we had summer last week and now we have winter again. Unlike other parts of Canada, our rainfall is very localized and most of it's lost to evapotranspiration, which this picture illustrates. So most of our runoff occurs during snowmelt when soils are frozen and can't react with phosphorus. So this is very different from, say, coastal Canada, BC, or Eastern Canada. And so we need to understand on the prairies how phosphorus moves because that's how we can manage it. So I'm going to talk about a study where we characterize physical forms, dissolved particulate. This is one snowmelt event, and we compared fertilized cropland versus pasture controls. So these were summer grazed for about 2 weeks. And then we also looked at management where we had infield winter bare grazing, where cattle were fed on straw, or where we applied manure from barns. These were matched to the same phosphorus application rate, and these were compared to these controls. We went out there in snowmelt, my postdoc here working. This box contains automated sampling equipment. It collected runoff every hour into bottles. We took those back to the lab. These flumes here were piped where the water came in. And just to show you, this is the pasture control compared to the bale grazing, which is kind of interesting all by itself. Anyway, we took those in the lab. So these are the bottles from this machine. We filtered them and then we analyzed them for all sorts of things, carbon, you name it, we've done it. I'm just going to talk about total phosphorus here. So this is the snowmelt. This is converted to account for flow rates. So because flow is different times, but these are, you can see over the 4 days of the runoff event. This is the pasture, the dark one here. This is the cropland. And so there weren't any significant interactions of date time treatment, but cropland always had more dissolved phosphorus, which was about, it said less than 1 milligram of phosphorus per liter. But when we look at particulate, that changes. The pastures release, but the concentration is almost a factor, 10 times lower. So most of the phosphorus is moving as dissolved phosphorus, and there were differences depending on whether it was from pasture. This partly is because in cropland, crops are harvested. There is phosphorus left in the standing stubble, but there's not much, whereas at pastures can actually have green plants at the time of freezing, and that's going to release different nutrients. Looking at bale grazing, this is what we showed. This dark one here is the control I showed in the previous slide. And this changes things a lot, whether you're bale grazing, spreading manure. As you can see, there's a lot manure spreading, a lot more dissolved phosphorus. These are 4 times higher concentrations than we saw in the last slide. Particulate, on the other hand, though, bale grazing releases a lot more particulates, and again, 10 times higher than what we saw. This is because the cattle, it's partly from the hay that you're putting out there that releases particulates, and because animals are moving and breaking up any frozen plant material. We also did some work with Jeff Shano and students at the University of Saskatchewan. They brought samples into the lab to look at runoff, and this was looking at different fertilizer placements. So we had controls, seed place phosphorus, deep banded phosphorus, broadcast and incorporated, or just broadcast at different concentrations. So this is with conventional tillage, and this is no-till. And for this, we just looked at concentration of dissolved phosphorus only. And whereas this is, we looked at both inorganic, organic, and dissolved and particulate. But what's really clear is where you put your phosphorus affects how it's going to leave the land in the spring. In snowmelt. It makes a pretty big difference. And particularly, and the higher contrast, higher concentrations of broadcast, the more you're going to lose. So again, effects, but tillage, of course, makes a big difference as well. So in summary, loss depends on land use type, cropland versus pasture, on fertilization, chemical fertilizers, bale grazing, manures, depends on fertilizer application rate and depth. It's really important though, phosphorus loss triggers algal blooms, so let's keep it on the land where we want it for our crops. So again, publications. So that brings me to the end. In the summary, phosphorus is an essential nutrient for all organisms, and it exists in different soils. So soil test P is just measuring phosphate, but there's all sorts of other phosphorus forms, and these are really important in terms of cycling. Land management affects phosphorus forms, it affects concentrations, forms, and how they are, where they are placed in the soil, and how, of course, they're going to be lost from the soil. Again, phosphorus cycling is really, really complicated. And most of what we look at is a very overly simplified portion of it. On the other hand, though, we need to have it overly simplified. There's no way we could test all of these things in every soil for every farm for anywhere. But we also need the research that I do to help inform those decisions and tell us what those soil tests mean. The other thing I'm going to come back to is stressing the depth of soil sampling is really important. Phosphorus is always higher at the surface and it doesn't move downward in the soil on its own. That's very different from nitrogen. Phosphorus doesn't move. And so if you're putting your fertilizer here, it's going to stay here for the most part. And if you soil sample here, it's going to show you that your fertilizer's here. But if you soil and if separate it from the lower depths, but if you soil sample and mix those together, your fertilizer test will always tell you to add fertilizer because it's going to reduce that phosphorus. So the key message from this is soil sample at the depth of placement. I acknowledge I work on land work, live and work on treaty forest territory. I also want to thank the many people who helped me with this work and various project funders. And that is the end.
Okay, well, thank you very much. We really appreciate the presentation, and we have probably about 10-15 minutes if we have any questions. So if any of you have something that you'd like to ask, you can either raise your hand and Jordan and I will be monitoring that. I see one from Brian Weeb coming in. The other one would be you could put a question in the chat if you're a little bit more shy. If you want to turn your cameras on, then we can see you. That would be fantastic as well. But go ahead, Brian.
Hi, Barbara. That was really great. You covered an amazing amount of material. I'm going to
really
go
fast.
through that recording a little more slowly. But your last point is the one that I was planning to ask, because this question has been giving me, well, not nightmares, but I have been struggling with this for quite some time. And this is probably the perfect audience for it. You mentioned sampling where you're putting the fertilizer.
Yes.
How do you do that when you've got a no-till situation and you've got the fertilizer banded? So you don't just have the vertical stratification, but you also have a horizontal stratification. And how do you sample so that you have a soil test that means something? Because I had an agronomist a few years ago, or a producer say his agronomist told him to ignore the soil test results for phosphorus because they don't mean anything. which basically told me they weren't sampling where they should have been sampling.
Yes.
But how do you sample where you should when you've got that sort of stuff
Again,
going on?
presumably, if you have a sense of where you band it, so depending if you're soil sampling, when you can still, for example, see crop rows or something like that, that can tell you where you should be sampling. But you probably want to sample where with and without banding, and maybe potentially send those for analysis separately, right? Or you're sampling by depth and just sampling all over the place with banded or where with or without banding. But again, dividing it by depth to get some sort of average, because presumably you're banding in different locations each year as you, it's not going to be, so it may average itself out, and it may not make sense to go exactly where you've banded as long as you take that depth of fertilizer placement into account.
Thanks.
All right, thank you so much, Brian. Are there any other questions? You have a group of people in the room, including our guest speaker today. So if you want to, ask a question to the audience, you can do that too. You can all see who's in the group, I believe, by looking at the people who are joining us. Does anyone, any other questions? I have a few that I can ask. So we already talked about, I had a question about the soil test, but that's great that Brian asked that one. Where do you see like one key takeaway for ag retailers when they're talking with their growers? Is there a misconception that you have, or is there that they might have, or growers might have, that you could dispel sort of a myth right now or anything like that?
Well, again, we need to, the 4R approach to fertilization is really key. It's the right source at the right rate at the right placement. The problem is if your soil sampling is not correct, and I think for phosphorus, we tend to soil sample for nitrogen far more than we sample for phosphorus, and that's an issue. So you're probably not getting the right form at the right place at the right time, right? Because we don't sample for the right nutrient, but it also comes back to what I mentioned about acidification, is you don't have to sample at all the depths every single year, but maybe every few years it should be done to monitor other nutrients besides nitrogen, to see if you're building up your phosphorus at levels that you don't need, but also to understand if you're getting that acidification, because that's with phosphorus. Oh, the publication, it is listed in there. I can send a link for that as well, for that publication. It just came out in the Canadian Journal of Soil Science, and it's open access. That was that question. Anyway, though, this becomes really important is yeah, right? Following the guidelines. The guidelines are there. It's just people don't give phosphorus a whole lot of respect in a lot of cases until they have a problem, because it's causing runoff.
Okay, I see Jacqueline. Do you want to join in on the conversation with a question?
Yes, please. So, in your long-term study, in the spot where we were not, specifically, we weren't seeing significant yield changes when we weren't applying phosphorus.
Yes.
And so I've seen with clients in the past and things like that, they have been pulling back their phosphorus because of the cost of fertilizer in the last few years and things like that. And when I tell them, hey, we don't want to do that, we are depleting our legacy phosphorus reserves, but they're like, oh, I'm still getting the same yield. What would be your response to that?
Again, are you measuring the right depths to understand if you are depleting the legacy? And again, how much legacy phosphorus do you need as a reserve? A lot of cases, we've gone past build and maintain. We're still trying to bring it down to a level, and then we get to build and maintain. So there's no one answer for that, because it really depends on how much phosphorus is in there, and whether, if they've sampled it at the right depths to understand, is it all sitting at the top of the soil, or is it, so there's, again, their soil tests keep coming back saying you need it, but they really don't, because the surface is really high. And again, it depends on the region that you're in. We have long-term plots on that project that have never received soil phosphorus, and they are still not showing that they would probably be responsive to phosphorus. The Olsen P is 10 ppm. But because we return, they're tilled and we return the crop residue, they've just got a maintenance amount. So yeah, we could probably boost our yields up significantly by adding phosphorus. So there's a big difference between need and what your goals are as well. So that my answer to this again is there's probably not a single answer to a client. It needs to be based on sound testing at the right depths and to understand what's going on in their soils.
Jacqueline, do you have any other further questions to add, or I see Vikram's hand, but is there anyone else want to add anything to that specific topic? Or your others to the audience, if you've dealt with this with your growers, how did you handle it? What kind of responses did you have to the question Jacqueline asked? No? Okay. So we'll go over to you, Vikram. Sorry. I just thought I'd sort of try to engage in a conversation there, but go ahead, Vikram.
Hi. I have tried out mycorrhiza, but I did not get very good success. Do you have experience with mycorrhiza and some companies which are promoting it? Which species work better in different soils?
Again, there's not really one answer to that because every soil, different plants have different mycorrhizal, whether you're adding it and it can compete against the natural mycorrhizae that are already there. And it's not something I actually study, so I'm going to be very cautious about answering that as well. But I guess, yeah, it requires some knowledge about if the companies are selling it, it's probably good to get some data for the specific soils in the specific region where you're going to try to, and crops in that region, to understand the effectiveness. And hopefully they've done some testing.
All right, we have one more. So Vikram, does that wrap up the question that you had?
Yeah. She indicated that she did not have much work done in this field.
Okay. It's outside of my area.
Yeah. Okay. Okay.
So
great.
I'll leave it at that.
Okay. Nope. That's awesome. I saw another question. Go ahead. Nair, is that, go ahead. We can't hear you. Is your microphone on?
Hi. So I was going to reply to Vikram's. I've done a little bit of work on mycorrhizae. So one, they would work better in low phosphorus soils because that's the association they form to extract the phosphorus from the micropores. So I agree that you have to try different species of mycorrhizae in the soil and how it works with the plants. One of the companies that I have worked in the past was Premier Tech. So they have got good products that work on most of the crops, and they can provide good results. So just a comment.
No, that's awesome. Thank you. Any other comments or questions around this topic from anyone else out there or joining us? I see Benjamin. There we go.
Hi, Barb. Great talk. I really enjoyed that. No, didn't want to take too much of the question time because I'd ask you other ways. I'm kind of curious about struvite and if that is getting more common and what the general feeling is on availability of a nutrient source. You know, the city of Edmonton, they've installed in their wastewater treatment plant a plant to recover that, and of course that's a important part of the circular economy and all that kind of stuff, and putting it back to the agricultural land. Is that becoming available for producers, or what's the status of struvite?
I honestly don't know. I'm about to start a study where we've got soils at different pHs. We've acidified and not, to see. Struvite is one of the products we're going to test. We want to look at availability. We're going to compare it to liquid ammonium polyphosphates and MAP, and sort of, again, to look at the effects. I was just involved with a study in Spain, of all places. Again, it was quite available. The two crops, it was certainly available in fairly higher pH soils. Again, it all comes back to cost. It comes back to availability of the product, but then whether the phosphate is available, it certainly seems to be, but I don't know how compete. And this is outside of my area of expertise. I don't, but it certainly, I think reusing waste and encouraging reusing waste. Phosphorus is fertilizers it's mined in countries like Africa, or it's mined in Northern Africa and countries like Morocco or other areas where there's geopolitical implications. And then we move rocks from Africa to Canada to produce fertilizers. And you got to think that maybe and then we consume it and it becomes a waste that we throw in the trash. Literally, sewage sludge gets in Vancouver, gets dumped into, they don't reuse it. You got to think we should be trying to capture that somehow. But again, producers, it's going to depend on the value and the cost and what you're growing.
Thank you.
All right, just sort of a little bit about phosphorus stewardship. What do you think the biggest opportunity is for the ag retailers to take a lead in that phosphorus stewardship in the work they do as that trusted advisor to the farmers?
Well, to really, again, the soil testing becomes the number one thing I stress, because that's going to affect not just where you're making sure you're putting it at the right amount, the right place, all the 4 R's, even looking at the product. One of the things we're showing, well, you saw that with our long-term studies. When we stopped adding MAP, the pH went back up. So that's not even considering the nitrogen coming in from anything else. That was just MAP. So again, if acidification on the prairies, and I just put a link in the chat to the paper, you should be able to look online with that DOI link, and it's open access, so anybody can get access to that paper. Maybe we need to be shifting. Although triple superphosphate is considered, it is acidic as itself, it's used, it may not be acidifying. There's a difference between applying an acidic fertilizer and an acidifying fertilizer. And it also contains calcium. And one of the issues with soil acidification is the loss of cations like calcium. Or maybe we need to be looking at other things. But I think on the prairies, we need to start paying attention to some of this, because once things get to a certain point, and they're seeing this in Montana and North Dakota, you get diseases, it affects crop diseases. It affects things, soil pH is a huge issue. And so from some of the things is to understand, or maybe bring in other products. At the co-op in town here, I can't buy triple superphosphate. I can order it, but I can't buy it here. I can't buy lime in Saskatchewan, but that's a whole other story too, because the assumption is we don't need it, but that's not true. And if consumers start demanding it, the products start coming in, right?
Okay, awesome, thank you. In the question and answer, there's 2 questions. So if you just want to take a look, there's one from Jan and one from Vikram. We probably have time for one more. Can you handle either of those? Do you see them there?
They're in the Q&A. Okay, sorry.
No.
I will click on there. Have I done any research on high effect? I don't study phosphorus fertilizer.
Okay.
So I study everything but the fertilizer, because I'm interested in everything else.
Okay, and then the one from Vikram.
Realistic potential of mycorrhizal uptake. Are there certain species that there are? My master's degree was actually looking at mycorrhizal fungi and winter wheat in British Columbia, which is not a big wheat growing region, but anyway. And the problem is they can be very specific, and they change potential of them as a commercial source. Again, they should have potential. The problem is, can we apply them that will outcompete natural fungi that are already there in the soils? And again, if the phosphorus is, my soils in British Columbia had a lot of phosphorus. The mycorrhizae became important when they were drought stressed, because phosphorus is diffusion limited. So there are high phosphorus concentrations, but it couldn't move. And so the mycorrhizae were extending the roots. So there are other factors that could be in play for things like but I think making sure that we don't understand the biological components particularly well for phosphorus cycling at all. We have very specific organisms for nitrogen cycling, but we don't for phosphorus. But I actually think there is more specificity. We've just never been at a point where, because phosphorus concentrations are quite low compared to nitrogen or carbon, and even having the techniques, the genetic techniques or things to identify the phosphorus, the organisms involved in phosphorus cycling. Does that answer your question?
Yeah, well, I'm not sure if that answers my question, but as long as it answers others, that's fantastic. There's one comment I want everyone to take a look at in the chat that Benjamin put in about echoing and reinforcing Barb's early comments. So you might want to read through that one as well. Is there any dialogue or anything you want to add to that, Benjamin? Because we'll be wrapping up shortly.
No, I just wanted to point out, Barb commented on this early on, and the middle number on your fertilizer bag is reported as P2O5, and we have as much P2O5 in that material as what Barb showed in her initial slides. There is none, but it's a convention of reporting it. But when when you're looking up how much phosphorus should I put on my pea crop or something, they will say, well, you need this much actual P. And I look at it and you compare it to the research literature that's done right, and it is not actual P. It's actual P2O5. So you can, and the error is 2.3 times. So you have to be careful as an agronomist, I would think, and check those calculations.
And to me, why are we doing those calculations? As we said, the labs is doing the test for your phosphorus is going to be doing it as phosphorus, as P, not P2. They then convert it to P2O5 to tell the farmer, who then goes to the fertilizer, which is, again, phosphorus analyzed not as P2O5 because you can't. It doesn't exist. They analyze it as phosphorus and then sell it to you as P2O5. Let's skip that conversion and just use phosphorus. And again, if people are used, I know people say, well, everyone's used to talking P2O5. Fine, but why can't we just report it on the bags both ways? And much like I'm old enough to remember when we switched to kilometers in the metric system, we just over time gradually reported both and got used to it. And we can't say we have to use P2O5 because that's what everyone's used to. Yes, but it's so meaningless. And it adds this extra confusion because some people, again, are you converting or not? And why are we converting in the first place?
All right, I'm in a bit of a coughing fit. So Jordan, I'm not sure if you're there and you can wrap up.
I was just, well, what I was going to say is there's one last question that just popped up. If you want to go into the Q&A section, Dawson posted something in there.
Let me see. Oh, I'm going back and forth between the Q&A and the chat. Any way that you research to open up the label P, and again, that's sort of what we're interested in. How, again, although I don't necessarily work at looking at how to open it up, what we're seeing is with things like the shift with pH change, that acidifying fertilizers were a map were used initially on the prairies in high pH soils, so over pH 8, because they would help make that phosphorus more available. The problem is continuing to use those as the pH drops is making them less available. It's a bit of and also just we know absolutely nothing about crop residues that return to the soils. Our nitrogen tests will often account for them, but phosphorus tests do not. We completely ignore it. But as I said, we have plots that have never received phosphorus. The only phosphate they get is from the residues, and it's keeping them going. They're not deficient. They would be responsive to fertilizers, but they're not deficient from 1967, which tells you something about that it's there. They were probably deficient in early years, and then it's built up into the organic phosphorus on the surface. So there's a lot we don't understand, and our soil tests to simplify things, which again, we have to simplify things for ease. But we have to remember that there's a lot beyond those numbers, and to try to think of the other factors.
Perfect. And with that, Dr. Barbara Cade-Minan. We appreciate your time today, and everyone that joined us, thank you for the questions. Great interactions. Don't forget, next week, Thursday, we do have another session, again, starring one of our Ag and Ag Food Canada researchers. Again, appreciate everyone for showing up and supporting the industry. We will see you again next week. Thank you all.
Thank you.